cloud. Cool. So yeah, thanks again, Ciarán and Francesca, for a wonderful talk today at the BBI seminar. I've got some kind of more general questions around sort of your roots in science and what got you interested in the sorts of work that you're doing. I'm going to kick things off. I'm going to start with Francesca. So what got you started in science and eventually led to you actually working within kind of synthetic biology as a field?
So, okay, so if I can be honest, I have never been one of those types that will tell you, like, when I was in kindergarten, I got fascinated by, like, you know, I've always been interested in a lot of things. And I wanted to be a singer, I played the saxophone, you know what I mean? Like, I really wanted to do different things. I wanted to be a journalist. And I think that without realizing it, I had love for science that came from my brother. So my older brother is graduated in physics, and he's always told me, you know, stories about Richard Feynman, you know, Show Your Joking, Mr. Feynman, all these books, right? So there's been always a love for science within me, and that probably why, when I was randomly choosing like courses, like university courses, I selected journalism, but I also tried biotechnologies at the University of Bologna. And while I was doing it, then I realized that actually I was really loving it. And so I stayed there. I didn't go away, and I continued. I got my graduation. And then after that, again, I thought, okay, let me find a job. And I ended up meeting a professor at the University of Bologna that was looking for someone to be an instructor for iGEM, for the synthetic biology student competition. And he hired me for 6 months to just really work with the students on iGEM. And I loved it. I really liked it. So I got into the synthetic biology area because I really liked it. And then he had a PhD position available, and that was it. Then I started, and after that I really didn't want to change. Yeah.
What about you, Ciarán? Is it a similar sort of random route, or did you always want to be a scientist or something?
No, it's pretty random as well. Really good teachers in school. I think I wanted to do medicine originally, then realized I'd be an awful doctor and probably didn't like blood that much and these kind of things. But along the way, I think genetics was a thing I really liked. So I got into that kind of stuff when you're doing your school stuff. Went to the genetics at Glasgow Uni. And when I was there, it was very human medical genetics, which I wasn't so keen on. And so I was kind of thinking, oh, how do I use what I'm doing to do something I'm more interested in? And then you touched upon it in the talk, in the intro to the talk. I got a chance to go to Ecuador for 2 summers, for like 3-4 months at a time. We were in the Amazon, and it was bizarrely like to do with snakes and lizards and frogs and doing like a census of what was there. So just to try and what was happening was this tribe in the Amazon that basically were under a lot of pressure from logging companies, oil companies, to essentially sell the land. And nearby tribes had done it, and essentially they all live in shanty towns, and just it's a mess, unfortunately. And so this was a little bit of that, so we could do a little bit of like genotyping of what was there, and so I thought, okay, and it's kind of conservation plus the genetics. And then heard about this thing called synthetic biology. And when I graduated, I went traveling for a bit, and just came across this idea of synthetic biology. Really liked the idea of it. Applied for a few PhDs, and from a internet cafe in, I think, Melbourne, where there was a really nice owner who was hushing people out of the way to allow me to do this interview. I managed to get an interview for Frank and go to Dundee, and so went in from there. And again, iGEM, supervised the team after the first year of iGEM, and that really gets you into it, you know. So yeah, it was cool.
Yeah, I think iGEM's a common theme with a lot of people. I think it really was like a crash course in what's synthetic biology, and also it gave you the opportunity to kind of define it yourself a little bit in terms of what you ended up doing, which I think is awesome. Which makes a big difference. So my next question is probably going to touch upon the sorts of things you spoke about in your talks, because both of you are battling with demons that is biology not doing what it's meant to do. So what do you think, and this can be related to the things you're working on, but it can also be more broadly as well, are the biggest challenges and hurdles that we're facing currently in the field, that if we could get rid of them, we'd have a home run in terms of actually taking things forward? So yeah, Francesca, what are your thoughts on the kind of biggest challenges?
So I think that, okay, probably I'm not saying anything new for people that work with mammalian cells. What I'm finding compared to my work with bacterial cells is really that if you don't have a lot of money, in a sense, like maybe at Berkeley Lights, they have the way to screen like thousands of cell clones at the same time, right? But if you're a normal like lecturer like me, where you have your pot of money, whatever, like the fact, for example, that even simple things like library generation and screening in mammalian cells is not as straightforward as it is in bacteria. You just don't throw the library members in the cells and you screen it, right? So you have to build them, then you have to transfect them. So the complexity, like the lack of throughput, I would say, in mammalian cells, the instability of the integrations. So it's quite challenging because you integrate and then after a few months, that thing is silenced. And of course, things are improving, but there's a lot, I think, that needs to be done there. And yeah, the throughput of the experiments and the characterization, that is not really very straightforward. And as I mentioned in my talk, is the difference in the different cell lines, right? So you have a protocol for transfection that works for ACK, it doesn't work for CHO, it doesn't work for the other. So every time you have to re-optimize. So really, I think a lack of standardization in the protocols. It's very, very basic, but I think makes a difference in the everyday life of researchers.
Yeah, I guess lead on from that. I'm wondering, do you think though? So I've spoken to a lot of people, and you get kind of two camps. One is that standardization is a waste of time. If you try and do it, biology's got too many knobs and things that you can change, and it's too diverse. You just can't do that. And other people, I think, coming from more your perspective, where you're like, well, no, you have to have something that you understand to start from. Sort of, what would you say to people that are sort of saying, well, actually, no, biology is too diverse. We should be embracing that diversity rather than kind of trying to get rid of it in a way.
I think it would be a bit in the middle, where I go for a characterized non-standardization in a way. So like in a way, okay, we say it's not standardized. We don't care. I don't think that is the way to go, because if you characterize why, or the context why is not standardized, you know what I mean? Like, and then you can describe it in a formal way. That is already a way to go for kind of a standardization of like setting the rules, understanding the context where things hold and where they don't, right? And that is a way to form like maybe mathematical formalization, not a unique law, maybe, probably will not get there, but there may be contexts where that can be applied. So I would be more inclined to go for this approach.
That sounds like a good compromise in some sense in terms of those things as well, which is good. Ciarán, what about you? What do you feel? What annoys you in the morning when you get into your lab and you're like, why is this not working in some sense?
Oh, I mean, repetitive sequences. They're the thing that annoy me the most. I still like, I mean, other companies are tackling this stuff, but repetitive sequences are still a huge problem, I think, that we haven't got on top of, in terms of we have this power of DNA synthesis, but every company has the different sales pitches and kind of different technologies and stuff, and are overcoming a lot of it. But I still think we're not there yet to just design order a fragment that we want, so that's the first thing. Like, for example, I talked about in the talk about the putting constraints or compromises around these sequences. We have to do that because for, let's say, 10 of those cytochrome P450s that we tried to make, we looked at 10 of them, and 4 of them passed IDT without any compromises, and I think another 4 didn't pass. So that was our arbitrary choice of which ones we're going to actually produce, right? So that's the first thing. I think, like, that kind of tackling that in some way that doesn't involve subcloning, right? So I don't think it can't involve subcloning. It has to be like a proper synthesis kind of chemical step. And then the other thing, I agree with Francesca, finding the balance between like a framework and allowing then the evolution and randomness of biology to take over is really important. Some of the kind of hurdles, a lot of the stuff that is done in academia versus industry, I still think we're kind of we haven't found the right balance and direction of what should be done almost by industry and what should be done, let's say, by academia, because there are some companies, as you know, like Ginkgo and these large companies, massive foundries, their high throughput kind of brute force approach, they can, if you have pathway X, like I talked about a pathway today, big pathway today, they can solve that with brute force. right? And I don't think necessarily then, putting in lots of different foundries in the UK that are often, unfortunately, still underutilized or not very open, in my opinion, to the community. Using brute force at the academic level for those kind of, I want to make pathway X, I don't think it's necessarily the right approach. And I've spoken to a few people who have foundries, and they say they actually struggle with this a lot, because they're trying to make chemical X and they're trying to optimize it and they're trying to increase the yield. In my opinion, that's kind of not really an academic exercise. There's ways you study what happens during that. I think that's really important. But I think then putting in new ways of doing biology, new ways of building biology, new ways of doing completely new things in biology is more where I think the academic stuff. So, and I sometimes feel like that can be actually a hurdle, that we focus too much on these big, amazing success stories of big pathways that have taken lots of time and brute force. And I think we sometimes neglect the more nuanced, interesting type of projects.
Yeah. What do you think? So I think that's a really interesting point to make that I hadn't actually thought about, because you often find also in terms of funding, academic funding, that there are some kind of cycles around things being more applied or then more basic research, and you switch between these different kind of modes. What do you think? Is there something that you think could happen that would, I don't know, make industry want to really take the reins on some of that? Because I guess it's not just a push, is that they should be wanting to pull this in-house, not just rely on academics to look at these things. What do you think's missing at the moment that would kind of allow that in some sense?
It's a really good point. I'm not too sure, because it's kind of the antibiotic challenge, isn't it? Like they don't necessarily have the time to tinker or like to invest. They have to deliver very quickly on short time scales. And that's where the argument that academics should do it is more interesting. But is it a great use of public money, for example, to have an academic working on something to be commercialized by a company? I don't know. Maybe not. So I don't know. It's a tricky one, and maybe there is no way of solving it. And this is the challenge that's always existed.
And well, they say whenever there are people involved, it's always messy. There isn't an optimal solution. You just try and do as best you can. So I think probably that's where we are, I think, anyways. Okay, so kind of change tack a little bit. So both of you are kind of working at the kind of forefront of synthetic biology, and you'll probably have some ideas of your own about where the field might go in the future. I'm just intrigued to know what you think the biggest impact of synthetic biology will be on, say, someone in the street in their everyday life in, say, 10 years' time. So, Francesca, is there something that you think that the world's going to be very different because of the sorts of research that you're doing at the moment?
Unfortunately, probably not because I'm doing it, because other people are doing it. Because I think that biomaterials, so bio-inspired materials, will have quite an impact. I think really that will be, but I'm not actually. Don't ask me why, but I'm not working on it. And another thing that I really like, and I think will have an impact in the long term, is like the Genome Project, right? So understanding more on the human genome. So not just because we want to add it as we want, but because we understand how it works and the role of specific areas or specific regions or specific sequences in the behavior of the human cells. And I think that will definitely impact the applications of what we do. So I think I would say these two things.
Cool. And Ciarán, what's your kind of sci-fi future look like? What's your dream tonight?
I share Francesca's comments about biomaterials. I think they're most immediate and medium term kind of probably applications of synthetic biology that can have a real impact on things. And I'm kind of this was an idea that came from Harrison. It's what inspired our kind of latest grant. But there's loads of other people like Tanya Tishart in the States and people like this that do really cool stuff on linking electronics with biology. And I think if we're going to start having kind of like, let's say, the Internet of Things with bacteria, for example, I think that would be a really cool thing to see. And I think there's enough people pushing for that at the moment that I think we'll start to see some progress towards that. And that plays into then like the idea of living buildings, and I think if you want to talk about, again, they're going to be smarter, kind of more responsive. It also probably plays into things like AI. So having living organisms mimic neurons and those kind of connections, which could be quite cool as well. I mean, there's plenty of funding that people are now starting to get in that area, and it's because I think people can see that it's a direction that we should be going in, and it's interesting. I'm not in that area either, so I don't know why we keep thinking about the ideas outside of our own.
Yeah, we are making a mistake somewhere, Ciarán, because we are not in the area.
You should be writing it. You should be writing grants in this area, see, and in the
Exactly.
future, you'll be defining what goes on. No, I agree. I think with both of you, I get super excited about sort of like new forms of architecture. So where it's not just a static brick, but it actually does something. It can adapt, it can change, it can respond. Like everything that biology does, that's really cool. That's embedding that into architecture and design. I think is super exciting. And Ciarán, you mentioned about this. Maybe you can say a little bit more about this biotechnology hub between Newcastle and Northumbria. That's about what's going on in that area.
Yeah, so there's three main areas, and I've got to try and remember them because, as I say, I'm hub adjacent, as I like to call myself. One is living materials. So how do you incorporate biology and have them respond to things as part of living materials? And that can be for things like bioremediation, or for like structural stress, responding to things like structural stress, or temperature control, climate control, that kind of thing, which I think is amazing. The other one is to do with products, so taking waste products from the house and producing interesting things from that. So a lot of it focuses around things like cellulose, where there's actually a bit of expertise at Northumbria and Newcastle in the kind of carbohydrate biosynthesis and enzyme pathways. And the last one is, as far as I can only describe it really as like the ecology and diversity of the living environment. So understanding and measuring and identifying what grows in certain environments that are part of the built environment, really at high throughput. So we have a real depth of sequencing facilities and technologies at Northumbria, that was recently used for the COVID sequencing. But their main motivation for setting that up before the COVID was part of this measuring what's in the living environment. Yeah.
Cool, awesome. Okay, so the last question, and I'll let you guys run away, is one of the reasons for making these videos is to sort of help maybe some of the early career or younger researchers kind of, I don't know, not make the mistakes that we've all made. So do you have any advice for someone thinking about starting off in a career in academia or science or industrial biotechnology that you wish you'd been given when you were starting? So Francesca, do you want to kick us off?
So there's one thing that I was told, actually, so I was lucky enough, but a bit later in my career, from a friend. I mean, so she told me, like, live for Monday mornings, not for Friday evenings. And that helped me a lot in a way, because that made me think, why do I have to be not happy to wake up on Monday mornings to go to work or to go to the lab and just wait for the weekend to arrive, right? So my life is just one life. I have one. And so I want to do something where I wake up on Monday because I really can't wait to go and do what I will do, right? And I think that if I am driven by that, then I really like avoid whatever doesn't give me the thrill to wake up on Monday mornings, and it makes me a happy person. And that's all I want, to be happy and to be fulfilled in a way from what I do, because that also allows me to be at service for the others or for the society, you know what I mean? Like, so there's kind of, so that I think is the thing that I have, that I was told and I pass on other people. Yeah.
That's great advice. Ciarán, what about yourself?
Well, it's hard to follow that one, isn't it? It's profound statements. No, Francesca is great for that stuff, by the way. If anyone is listening to this and wants advice from someone who's at the forefront of it, Francesca's, for me as well, a good sounding board on these things, career and stuff. And a couple of like less profound things, but like kind of go with the flow. You know, like your project may not work out exactly as you want it to go, but there's lots of interesting stuff that will probably be there if you have your eyes open and you're willing to kind of like pivot a little bit. Do try and focus on the projects that make you most excited and most interested in. And that relates to, you want to come in and really enjoy the projects you're working on. You're going to be the world's expert on your project, essentially, by the time your PhD or postdoc's finished. So really try and keep focus on those ones. You have to play the game a little bit with, you know, getting your papers and all these kind of things, but try to keep focus on what your kind of big vision and big picture is. Sometimes you don't have the opportunity, either it doesn't work out or you have to move around. That's really challenging as well. But I think, yeah, go with the flow, but still keep your focus on what makes you excited in the field that you're in. I think that's good motivating advice.
Excellent. Very profound advice all around. I think that's awesome. So yeah. So again, I just want to thank you both for a wonderful talk and also for taking some time out to sort of chat with me now. It's been really, really nice. And I can't wait to see what our futures hold with all the research you're doing. So it's great. So thanks again. Catch up soon in person.
Thanks.
Bye.
Bye.
Bye.